And please give your team passports. And also, actually, I have already introduced all teams. Biomatroshki from Tokyo Tech and Moscow State University, Watson Screams from Kuban State University, Russian Sushi from Tokyo Tech and Moscow State University, and Equilibrium from St. Petersburg State University. Yes. And today for the Science Fight 2, we have our jurors board, Mr. Alexander Lazukin, a researcher from Institute of Plant Physiology, Pushina, Dr. Tatiana Savchenko, the head of the laboratory from the Institute of Basic Biological Problems, Mr. Daniel Aladin, researcher from the Institute of Basic Biological Problems, Mr. Yegor Kosarev from our farm group of companies, from PR department, and from HR department, also from the branch of our farm company, Miss Anna Firago. Thanks. So we can proceed to captain's contest. It will be in the same format. Please, 4 captains come up somewhere here with a pen and small piece of paper. Yes? Yes, we have some time to wait for them. So I guess everyone should be ready for the captain's contest. Yes, please come up here. We'll have another question with a numerical answer, and the closest team will have an opportunity to choose firstly the line in the table. So the question is as follows: how many squats did the record holder do in 2 hours? Did you understand the question? In other words, what was the maximum for 2 hours number of deep squats? Squat is like this squat, this type of movement, yes. So, did you understand the question? How in 2 hours? No, human.
A human.
Yes, it was a record. I mean, it is the maximum recorded trial. So now you have 30 minutes. 30 seconds to come up with a decision. So the time is up. The correct answer is 3,311 squats in 2 hours. So the closest one was Team Equilibrium, yes, and you can choose the line and write it in. The second one was I guess yours. So you will write second. So you, in the first stage, you have one day and a half.
I think I'm lost.
No, the answer was no. Yes, next. There are 2 options left. Yes. So for the first stage, Russian sushi are going to do a report, but they have eternal rejection problem number 8, and they had already reported problem number 7. Yes, it is correct.
Yes.
And Team Equilibrium had already opposed problem number 7 again. Yes? So we can proceed to challenge. Problems that could not be challenged are problems number 7 and number 8. Yes, please, Team Equilibrium. And Team Russian Sushi will report.
Like to challenge is number 5.
Problem number 5. Also, you can pronounce the task's name to make it easier.
Looking forest.
Yes, to see the forest for the trees. Do you accept the challenge? Yes, it's first tactical rejection.
Then, but here problem number 6.
Oh, it is second rejection and time for Texas. Never late.
And then it will be number 3.
Yes, that's nice. So we have a time for preparation.
Я не буду указывать.
Не, ну в смысле, слайд.
Это что?
Нет, это микрофон. А это переключать слайд. Работает? Yes, you can start.
So, friends, Japanese countrymen, lend me your ears. I'm going to present you a problem about permafrost microorganisms. So, the permafrost is a frozen ground with a temperature called 0 degree and for 2 or more years. Permafrost even this permafrost hosts a number of a diversity of life, and these microbial members not only able to perform their basic metabolic functions, but they can also grow and divide. So there are 3 types of metabolic rates. The first type is the rate for growth. It refers to cells encountering in proper conditions to grow and to repair their molecular damage. The next rate is the rate for maintenance. It refers to cells that can only perform their metabolism, but they can't grow. And the second one is the rate for survival. It's the cells that are able to only repair their damage, and this condition is called dormancy. So, today I'm going to present this question in the terms of metagenomics. According to one work, researchers conduct metagenomics and 16 RNA sequences across Pleistocene, permafrost chronosequences from 90 to 33,000 years before past. So this method is involved the networks and pathways of genes, and I am going to mention some differences between functions of genes. The first one, according to environmental sensing. So genes involved in environmental sensing increased in all the samples. These genes encoded two-component regulatory system, which is involved in transfer of in temperature sensing, in protein misfolding, protein iron regulation, cell stress, osmolarity, oxygen limitation, cell envelope stress. Also, the genes that increased were involved in nutrient and resource sensing, and nearly every gene which encoded chemotaxis increased too. The second one interesting thing is about membrane transport in ancient microorganisms. So genes are we encoding secretion system increased in all the samples too. What were these systems? The first system secretes products into the extracellular milieu. The system number 4 is involved in nutrient acquisition. The system number 3 was responsible for interaction with eukaryotic cells, and the last one is the work of the second and third systems with the sister type 2 to excrete hydraulic enzymes. Another main is about cell envelope components. The genes encoding the synthesis of these components increased in all the samples. What we can see, fatty acids are responsible for fluidity. Lipopolysaccharides makes selectively permeable cells in gram-negative bacteria, and peptidoglycan can make a mesh-like cell wall which have a resistance to osmolarity, which is really important in permafrost. The next one interesting thing is about what kind of transporters uses in the cell walls. The genes increased in all the samples were about encoding ATP binding cassette transporters, which is involved in transfer of amino acid, peptides, osmoprotectants, stress molecules, and trace metals. Also, these genes encoded the transfer of lipopolysaccharides that form protective cell walls. Let's look on the metabolisms of different microorganisms in permafrost. The most metabolisms which were abundant are methionine metabolism, because it is involved in synthesis of stress molecules, and anaerobic catabolism of aromatic compounds. It's about, for example, the product from toluene and some other components, benzoate coenzyme A, which is one important compound of bacteria. Yes. So let's look on the results. So, in the permafrost, microorganisms depleted sugar metabolism and starch metabolism, because there is not sugar in the permafrost. And of course, about they use anaerobic metabolism and amino acid metabolism, aminocarbon metabolism, because there are a lot of carbon and nitrogen in the permafrost. Also, it's important for them to have fluidity to survive and chemotaxis, to have the chance to go to the place with high concentration of nutrients. What is more, microorganisms in these conditions just can't perform asexual production, because they don't have a lot of resources and energy. So they use horizontal gene transfer, and in the most of the samples, the amount of endospores increased because endospores is the improvement way of dormancy, something like this. Thank you for your attention.
Thanks a lot. So now it's time for clarifying questions from the opponent. So we have 2 minutes for your preparation.
Коронова.
If anybody didn't notice, the report took exactly 479 seconds. The following team, please. Also, please announce your name.
Should I stay here?
You probably should use microphone, but you can stand anywhere you want. But microphone is better.
This.
Yes.
Okay. Hello, my name again is Maxim Kuznetsov. I'm the captain of team Equilibrium. We are from St. Petersburg State University. So let me start my position. Generally, I really like this presentation because I heard a lot about factors and such things that are important in survival of some organisms in permafrost conditions. I do like the manner of presentation. It was clear. Actually, I have some clarifying questions. As I understood, we are talking about bacteria. Oh, yeah. We'd like to hear some more words about some other species, for example, nematodes or amoebae. Yeah, because in my view, they have different mechanisms of action to protect the organism from low temperatures. Actually, there was no words about what condition exactly we are looking at, because the temperatures, permafrost conditions can be different, and different mechanism can be applied. Actually, one more important detail is that the task states that you should have presented some factors that are preventive from survival. So in my view, there were generally majority of set was about factors that help the organism. We can discuss it later. I'm sure you know the factors that don't help. Actually, I would like to discuss in more detail what, in your view, the most important factors in the destruction of organisms. Like, is it crystals of ice forming in the cell, or is it other features? And we should also discuss. You were talking about genes and their action, but I would focus on how the system changes, because if you have an organism and it will become a freezed, generally speaking, it can be changed in gene actions and how it's done. Like that, and I don't completely understand what was that about hemataxis at the beginning, because it was like what so something has to be said about the factor that attracts something, if there is some kind of hemataxis, and why it's happening. I guess actually I do like the presentation, because many factors that I found in my research were presented, and they're presented properly. So I just can say that a lot was said, and that's fine. Thank you.
Thank you for your closing speech. Well, I would like to remind that a position is generally addressed and should be addressed to all of the audience. But in polemics, which will start soon, it is time to discuss anything you wish and to ask and answer any questions. So, wait a second, please. Now it's time for polemics, and both of you, each of you, has 5 minutes in it.
Now, firstly, I want to speak about what about changes which happens. So the goal of the method was to look how the community of bacteria and microorganisms changed with age. And because of it, we can see that in the 33 year there's firmicutes, it's the fungi. And in the 19 we can actinobacteria, proteobacteria, and bacteria. It's the majority of bacteria we have in this time. And I just want to see how this what kinds of features have this bacteria to help this bacteria to be successful in these harsh situations. So you will see for bacteria there are a lot of in this species we have a lot of gram-positive bacteria, and I think it's because of the fluidity and the chemotaxis. What is chemotaxis? Chemotaxis it's the ability to move. It's ability to move. Yes, and because there are in the permafrost there are lack of nutrients and resources, they have to move to do their low metabolic processes successfully. it's why pharmacos. It means that pharmacos have a better apparate to do this, so they have better chemotaxis apparate.
So they can easily find food for survival.
Yes, and the main
More
food,
easily.
it's like carbon and nitrogen from the biomass. Yeah.
And do they differ from another microorganisms?
Pharmacos differ really because they can form endospores. It's like dormancy but very low metabolic processes. Why these bacteria why a lot of bacteria can't to live in these conditions? It's because they become frozen and they have harm of because of water, crystals of water, and their DNA is prone to degradation. But when we speak about endospores, they have these endospores can repair their damage because it's a very good strategy to form endospores.
But what about chemotaxis? Like, as far as I know, endospores don't move. And we started with that. In your view, these microorganisms who survive from the very beginning, they have better mechanisms for chemotaxis. And my question was, could you clarify how it works? Like, do they differ from other organisms? Just if these species survived, like they have to have something different about chemotaxis.
Mm-hmm.
Or they just better defined in food.
They are better in. For example, fluidity. It's fluidity. It's because if they will be too harsh, they will.
You're talking about membranes.
Yes.
Yeah.
They will have damage from a lot of damage from ice. It's the feature that becomes more that increases with the age. One of the features.
Yeah.
Yes. And also, it's really interested about metabolism because the species in all the species has more aromatic hydrogenation degradation. It means that they use something like toluene, benzoate, 4-hydroxybenzoate in their metabolism, and we have a lot of this in the permafrost. This is another feature which increases.
Yeah, but you are talking now about how they gain other sources of food, like
yes.
Like they more effectively use what they have. Yeah, but what about membranes? What about their defense from freezing? Like they do something about it.
You mean what they do to prevent freezing?
Yeah.
Okay. Of course they do. They have. It depends on cell envelope components which I presented. It depends on peptidoglycan because it have resistance to osmolar pressure, and it's about osmolar pressure.
So it just becomes harder.
The cells have some improvements.
Yeah, but what kind of improvements?
So the beta apparate of peptidoglycan, lipopolysaccharide, and fatty acid chains.
Yeah, but what about liquids?
Liquids?
As I understand from your speech, it's
Fluidity.
Just becoming harder and harder and stronger and stronger the membrane.
No, the membrane not become
lighter. But do they have a special mechanism for removing water from inside of the
I think it's about hydraulic enzymes. Can I just
we know that water freezes, and if they want to survive, they
Yes.
simultaneously need water and they don't need water. And how they manage this problem?
I think that it's about secretion system number 2 because it increased hydrolytic enzymes, and these enzymes use water for in their work.
Yes, but I don't know, but I hear that they somehow produce some polysaccharides to make water like to alter the temperature of freezing. Do you think it's
Yes, maybe it is about selectively permeable protective barrier because so it can be selectively permeable for transfer water, for example.
Excuse me, the report is time for polemics that already finished, and the opponent has approximately 40 seconds. If he has 40 seconds, you are able to make any statements.
okay. I still think that the presentation was good. We all learned something from this. Yeah. And actually, during these polemics, we discussed some important features of this mechanism to allow these bacteria. What could be added is what other species do, like nematodes, because we were talking about bacteria, but no time for that. I think all these mechanisms do work in bacteria. That's all.
Thank you for your productive polemics. And now we can proceed to the review. You can have your seats here.
Thank you very much for the very wonderful and dynamic presentations and polemics. So it's a very interesting issue to discuss about permafrost because I believe we can learn a lot from those kind of organisms living there. And first I would like to start with the reporter. I think the reporter really give a clear explanation and some really detailed mechanisms about why organisms can survive in permafrost. But she gave a lot of mechanisms that maybe we think that if she tried to focus into several very crucial mechanisms, the presentation maybe could be a lot better. And we also think that it will be very good if she tried to make a table of comparison, for example, for organisms living in permafrost and organisms like normal microorganisms, so we can really distinguish the difference between two of them. And some of the features that is discussed, for example, chemotaxis or horizontal gene transfer, I think it might also present in the normal kind of microorganisms, so that maybe a little explain too much about that. And now I would like to move for the opponent. I think the opponent is very critical about the presentations, and he pointed a lot of things that is lacking from the presentations of reporter, which is very good. But as the chair of the session said, the opponent, when he gave his speech, is a little bit like two-sided only with him and the reporter. So it's maybe better if he could give it more to the audience as well. And then, although he's very critical, we think that there is no trial to eliminate the solution shortcomings based on the polemics that they do. However, we think our team think that the polemics was really good and dynamic. It's a pity that some of the very good and clear solutions from the reporter is only given in the polemics. For example, the benzene metabolism, something like that. If maybe you can explain a little bit more in your speech, I think it will make your speech even more wonderful. So that's all for my review speech. Thank you.
Thank you. Well, now it's time for questions from viewers. Yes, please.
The question was like 1,000 years of all about long-term survival. And if you will have two organisms which can survive for a long time, there is another group of organisms which can survive freezing, cycles of freezing, warming, freezing, warming. What do you expect the major differences?
You asked how to distinguish organisms between the chromosomes.
No, which can survive very short time freezing, and organisms which can live for a long time in this condition.
Their main features are
the time frame. So they should be able to live for a long time, not only short period of freezing. Survive for a long time. It should be different strategies to survive short periods of time or to live for years. What do you think would be the major difference between these two types of strategies of survival?
I think it depends on the chromosomes transformation processes. These type of bacteria and they don't need. How they face the evolution. So they just try to, or they can use some mechanisms of metabolism and making cells and proteins and these bacteria they have their mechanisms that can help them to survive and so they survive. And
yeah, I could try my best version. Actually, in my view, organisms that survive for 1,000 years, they try removing water from itself. Like you don't have water, you don't have problems, and you have time to do it because they can't be freezed like momentarily. Yeah. Yes, but I think that the difference with those who survive but with a rapid and short period of time of freezing is that they try to make some to alter the temperature point of freezing. So they do it making more polysaccharides, and they put some molecules in water to prevent freezing.
But there is not present. So these they should some energy, and if one has some energy, they should have some proper, some good metabolism. They have the evidence that will have them grow there, and to generate resources. For example, sugar and such metabolism decreases, but amino carbon and anaerobic catalyst of organic compounds decrease. And because why? Because there are in the form of carbon and nitrogen and aromatic compounds. Using these compounds, they have energy for survival, and if they survive, they have the time to have evolution, and this evolution will go in a good mass of proteins in the cell walls, which on the inside will book a water. And the first point is metabolism and the resistance, the resistance of the cell wall. Some kind of resistance of the cell wall.
Any other question? Yes, please.
For example, polysaccharides that many insects, for example, multiply in autumn, help them to freeze to a winter period. And there is evolution. Every winter they are freezing, and every spring they warm up. It's okay. It's evolution. Many times they make it. But for 10,000 years, is it evolution help them to freeze, or it's just luck?
Okay, I understand the question. So there are two theories. It can be evolution in the permafrost, or the bacteria just it was just the bacteria that encased in ice at the time of formation permafrost. But there is the fact that it is known that 19,000 before and 33,000 before it was just the same conditions in the world. It is documented. But nevertheless, these bacteria have differences in their genes, and suppose that it's because in 22,000 before past, the climate was changed radically. Yes. There is this branches. This fact is this branches posterior that it was just luck, and it is a branch to the theory that it was evolution.
Thank you for your answers. Any other questions? So thanks a lot for all participants of this day.
Now we can proceed to gradings, yes. Also, I would like to say that it's very nice that we have detailed discussion between participants and between jurors and discussion, but and it's the thing that we were looking for when designing problems. But nevertheless, I would like to ask jurors and participants to be a little bit more short in their questions and answers, because we don't have such time for such detailed discussion. So if everyone is ready, marks for the reporter. 7, 7, 8, 7, 6. Your grades for the opponent, please. 7, 8, 6, 7, 7. And for the reviewer. 4, 5, 4, 4, 4. I guess it is the first 5 for the review in this section for today. So, thanks a lot again for participants. And now we can proceed to the second stage, where the Team Equilibrium will do their report. Team by Matryoshka will make a challenge and do their position. What's the script? So, what's the script? We'll do the review. So, please just say what it was your eternal rejection. The second, and you have already reported problem number 1, and BioTournament had already proposed problem number 4. So, team BioTournament, you can not challenge for problems number 1, 2, and 4, and also problem number 3 because it was already recorded in the section. So please, you can start your challenge. Number 7. Do you accept? Problem number 7. Yes. Wait a second. Problem number 7 was rejected. And problem number 8. 8 is accepted. So the
Okay.
Назад.
Hi to everyone. My name is Daria Konechnikova. I'm from the team Equilibrium, and I'm glad to present you our solution for the case about carmine. So, to start with, I want to remind you the main goals that we have. So, we were needed to intensify the production of carmine or to create another red food pigment with those convenient, another red food pigment. So, and to start with, I would like to remind you that what is carmine. So it's an aluminum salt of carminic acid that is obtained from the dried bodies of scale insect, from Dactylopus coccus. That is feed on only one species of plant, which is cacti. So, also I would like to stop on the advantages and disadvantages of carmine. So the first advantages is high stability to heat and light, and also the high resistance to oxidation. As carmine acid is a natural pigment, so it has non-carcinogenicity and no toxicity to humans, even on skin and on ingestion. So that's why it's used really widespread in cosmetics, in food, and in pharmaceutical applications. So about the disadvantages, as I have already said, it's a natural pigment, so to make 1 kilo of carmine, you have to, you need really a lot of cochineals, and all of them usually collected by hand. So this extraction and purification and to make the carmine, it's really, really expensive. So the production is really expensive, and also it's vulnerable to weather conditions. And so all of those properties and all of those problems required us to create something new to intensify the production of carmine. And for this, first of all, we have to study the biosynthetic mechanisms of carmine exit. So first of all, carmine exit produced through the polyketide pathway, and this pathway starts with the cyclization of substrates, which are acetyl coenzyme A and malonyl coenzyme A, and this reaction catalyzed by the special ferments enzymes polyketide synthesized. And all of this process, this pathway is well described in bacterial and fungi and plants. And advantages for using plants is pigment stability and easy to extract those pigments from the plant. And also there are a lot of sources of plants that we can use for extract pigment of, such as seeds, leaf, and almost the whole plant. But microorganisms, they are easy to processing. They are inexpensive production, and also they have a short production period, so it's a rapid growth, and also it's independent from their weather conditions also. So that's why we decided to try the filamentous fungi, especially the Ascomycetes fungi, to produce pigment, because we know that they produce pigment as a secondary metabolized by polyketide pathway that we need. So then they also have a lot of substrate that we need, acetyl coenzyme A and also malonyl coenzyme A. So then they also have the necessary cellular infrastructure, especially the glucosyl transferase. It's a ferment that bound with the endoplasmic reticulum membrane. It's also necessary. Yes, I forgot on this slide to show this, because the last stage of biosynthesis of carminic acid is the adds to the intermediate product, the glucose unit, which is catalyzed by glucosyl transferase, which is bound to membrane of endoplasmic reticulum. And the last one is filamentous fungi. They have well-developed genetic engineering toolbox. All of those led us to design a semi-natural pathway of carminic acid in Ascomycetes fungi. So the first step, we have to select the potential polyketide pigment producers that will be safe for us. So it means that, the most safe species are Epicoccum, Penicillium, or Aspergillus. So it is the step one. The step two, so we have to eliminate another biosynthetic pathways which can contaminate the production of our main product, carminic acid. So then we move to the step three. We insert we take the polyketide synthesized encoding gene, for example, from bacteria, and insert in there our reference stain of fungi. So let us see what we have. So fungi have a lot of substrate, as you see, yes? Acetyl coenzyme A and malonyl coenzyme A. We insert the polyketide synthase gene. So now we have polyketide synthase. We also need the cyclase that is responsible for closing the rings. So we receive the intermediate product, it's flavocarmesic acid, And then we move to the step 4. We have to insert this last enzyme, T-glycosyltransferase, that is bound to endoplasmic reticulum membrane. And this ferment at the glucose unit, and that's we receive the carminic acid. And the last step, last but not the least, we have to create a special conditions for fungi, which will optimize the physiology of fungi, because we have to avoid the production of mycotoxins. And this is the last step. So we have to create the special conditions. The temperature, first of all. We know that fungi are mesophilic, so the optimum temperature for their growth is from 23 to 28 grades of Celsius. So also the special pH, it's exit pH. So also we know...
Excuse me. You have to proceed to conclusions.
Yes, okay. So as you can see on the slide, there are a lot of different conditions, such as the temperature, oxygen, light, and also the composition of the medium. And you have to do all of them just to escape from of the production of mycotoxins. Thank you for your attention.
Thanks a lot. Now it's time for clarifying questions, if you have any. Do you have any clarifying questions? No, so we have 2 minutes for preparation for the exposition.
Well, as you remember, my name is Alexandra. Thank you. I want to thank you for your presentation. I think it was great. Well, now let's start the exposition. So, I think there was good introduction. So the reporter said about advantages and disadvantages of the process of production of carmine nowadays, and about carmine, but the reporter didn't mention another colorants, maybe another colorants, not only carmine, so we can just product another chemical compounds with red color, maybe, yeah? And so maybe it will be it will be cheaper, for example, and easily. So it was pronounced that we don't have any problems with carmine. I mean, like diseases, and carmine is not toxic compound, but there are people with allergy of carmine, something like people can have something like dermatitis and other. Well, and the solution is to use fungus and to product microorganisms and to product carmine by microorganisms. But it's we can do it in laboratory. But how about this process? How about the industrial process? So how to do it in factory? I mean, so we need to product a lot of kilograms of carmine, and how to do it in factory with economical profit, and how to do it with fungus in factory. I mean, scaling. I mean, this process in factory will have a lot of problems. So, will it be possible to make it to product it by fungus? Is so to product it. To product a lot of carmine. Well, and what about enzymes? The processes includes not only two enzymes. So the enzyme, it's like it includes a complex of enzymes. It has about 8 steps, so 8 enzymes. And 8 unique enzymes, and we have to make 8 new protein. We have to make 2 fungi to product 8 new proteins, not only 2. So it's quite difficult, and there is such problem as metabolic burden or overexpression. So if we want to express a lot of new proteins in a cell of fungi, it is difficult, because it's like it can cause an inhibition of some enzymes. Well, and one second, please. I think that's all that I wanted to say. Thank you.
Thank you for your project. So it's time for polemics, and each of you will have five times, and then you can use another mic. If you want to have a timeout, you can. Yes, please. reporting team, please. So, you may start.
Yeah, I would like to thank you for your questions. So you asked me about another colorants, and if probably it's easy to production another colorant. So first of all, I said about others pigments. For example, from plants, I said about carotenoids. Yes, it's not all of their red colorants, but I mentioned that. So, but yes, of course, there are a lot of different red pigments and natural pigments and synthesized pigments. Yes, I also wanted to ask you which colors you mean, natural or synthesized, because there are some differences, of course. So, and yeah, you also, and the main, the main goals of this of our case was to intensify the production of carminic acid, of carmine. So that's why I answered this question. It was my solution.
Yeah, thank you. I understand you. I mean, maybe there are another colors which are easy, which we can easily product. So synthesized or natural is not so important, but something that does not need hand, something which does not need so a lot of work. Something easy.
Like human, you mean?
Yeah.
Oh, okay. Yes, you're right. There are a lot of synthesized red pigments, but they are not so stable, and they are cheap, but they're not stable, they are not natural, and also they can cause a lot of side effects, and they might be very cancerogenic. And so, yes, it might be a lot of side effects. That's why carmine is it's quite good. It's natural, it's good. And about the allergies. Yes, I mentioned it's natural pigment. A lot of biological pigments and stuff might cause allergy. That's okay. It might be, but actually, I have read a lot of articles about the physical and biochemical features of carmine. There are not so many reporters about their allergies, so it's quite non-allergy. Yes, and
so you read statistics about it?
No, it wasn't.
Of allergy?
It wasn't a statistic, but I have read the articles where they have been said that it's not really allergenic. Yeah. It's not statistic. Okay. No. And also about the industrial process. You asked me about the fungi factory and problems and etc. So, microbial production. This is the 21st century. It's really, yes, it might be, and yes, it's not really expensive comparing the production of carmine by the scale insects. By the handle work of human, it's much more expensive.
Yeah.
And also, yes.
Sorry, I understand it. I mean, what about stability of strains with 8 milliproteins? So, how they will be good at industriality?
You mean the species or what? The species, the species of fungi, is it stable?
Yeah.
Yes. If
stable with 8 milliproteins, I mean, will they product it for a long time or just for a short time? So we know that it's a problem.
Yes, but if you insert special genes in the DNA of fungi, so it might work quite good. And even with those eight proteins, yeah. And I also wanted to add that yes, you are right that the biosynthesis of carminic acid is much more complicated. But I decided that it's not the main thing to discuss in our solution. I mean, I wanted to show you how to insert the main genes in fungi, and that it will create special conditions, and it will work. It will produce a lot of product that we need, desirable product.
Yeah, and since it will work, I use it. It will work. It's okay, but these strains can be not stable. So
that's why we create special conditions that I also mentioned.
Yeah, but I think it's special conditions for better growing, not for stability.
Better
for production, for large production, but not for stability of strains.
Yes, but also, yeah, stability and growth. They're well. I think that not only for the well growth, but also for the stability, you have to save their natural conditions that the fungi used to have. Yeah, and also I really didn't get the question about inhibition of some enzymes. Could you please explain it?
Well, as I remember, there is so when we overexpress the complex of enzymes, it can be something like it can cause something like inhibition of work of one of the enzymes. So it's like.
So if I understand you correctly, so this complex of 8 proteins, and one of the proteins might inhibit the whole system, or what? But it's a natural system where it couldn't be possible.
If the amount of enzymes will be too large.
The amount of enzymes will be. Oh, so do you mean if we insert there okay, I guess I get it.
The short answer.
Yeah, I mean, I don't really get the question.
So if you will have a lot of a big amount of protein enzymes in the cell, a lot of enzymes, so it can cause something like inhibition of their work.
But we insert not the random enzymes. Yes, the short answer is we do not insert the random enzymes. We insert the special enzyme that will have an influence on the special pathway. So I think that's okay. There is no inhibition there.
Yeah, it's maybe okay. I just
it's maybe.
sorry to interrupt, but the reporter's time had already finished, and opponent has approximately 1 minute here.
Okay, thank you for answers. They were very good, I think, and as for me, it's a good solution, but the solution is a lot of problems, so it's not. I think it's normal, but it is normal, yeah. So I just want to say that it will be difficult to. It may be difficult to use this solution for production of the big amounts of carmine, so for big production.
No.
I think that's all.
Yes.
Thank you.
Yes, thank you for your polemics. And now it's time for the reviewer to come up to the stage.
Hey there, my name is Margaret, and the first one, I have some questions. There is a second presentation about coming today, and I noticed that we all have different quantities of needed insects. I have an information that for 1 kilogram of this dye is 200,000 insects required. Do you have any reference for this info?
200. Sorry, but the reviewer does not reply in polemics. Reviewer makes his reviewing statement, and that's all.
Okay, so thanks for interesting presentation of the solution. All was clear and easy to understanding. The presentation hasn't a slide with contacts and reference list. I have report was good structured, and I really like this solution. You showed us advantages and disadvantages of your solution. It's good for ecology and cheaper than classical extracting. I had noticed a mistake in opponent's speech when she asked a question that don't about the reporter's solution but about the alternatives. Opponent was perfect when she asked about industrial, how can we get it in the industrial scale, and it was really good. Polemic was amazing. All of the details was marked, and now we know more about this solution. Thank you for attention.
Thank you for your review. And now, please, any questions? Maybe someone will start producing termites tomorrow. Well, only short remark.
I just wanted to say that I don't have the list of references, but I have almost on the each slide I have the link, not one.
Yes, thank you. Any questions about the solution? Any questions to the opponent or reviewer? Thank you. So thank you all again. And let's proceed to the grading. Yes, your marks for the report. Ten, 9, 9, 7, 7 for the opposition. Eight, 8, 8, 6, 5 for the review. 3, 3, 2, 3, 3. So now we are going to have a break. I think we do not really need a big one. Yes, I offer that exactly 5:15 we will start the third stage.